I'm CJ Anderson, a postdoctoral fellow in the lab of Koti Ravichandran at the VIBU Ghent Center for Inflammation Research, and our group is obsessed with cell death. As you might know, our body is composed of tiny building blocks called cells. When a cell is too damaged, too old, or too sick, it will die. Roughly 200 billion cells in our bodies die on a daily basis. That means while you watch this 3-minute video, about 400 million of your cells will die. Now don't worry, these cells are replaced by healthy new ones by the process of cell division. Now in contrast to healthy cells, cancer cells no longer decide to die when they are damaged, but on the contrary, they rapidly divide until they form tumors. Now the fact that cancer cells divide so quickly makes them vulnerable to chemotherapy, as this type of therapy actually targets rapidly dividing cells and forces them to die. While chemotherapies can be tremendously effective at killing cancer cells, this doesn't come without a heavy price. Unfortunately, these chemical agents are unable to distinguish between a rapidly dividing cancer cell and a rapidly dividing healthy cell, like the healthy cells in our intestinal tract. This means that when we are trying to kill cancer cells anywhere in the body, chemotherapy inadvertently kills the cells in our intestine. This leads to some of the most well-known side effects of chemotherapy, such as nausea, vomiting, and diarrhea. And these side effects can be so severe that cancer patients often have to reduce the amount of chemotherapy they receive or even stop treatment altogether, which leads to ineffective killing of the cancer cells and regrowth of the tumors. Even further, chemotherapy can leave patients more likely to develop more frequent and more severe infections, but we don't understand how exactly. One thing we do know is that the trillions of tiny microbes in our gut, termed the microbiota, have an important role in this. Normally, this bacterial community is well balanced and actually assists in many essential functions of human health. In fact, the human body consists of more bacterial cells than it does human cells. However, chemotherapy can disrupt this balance, making harmful bacteria become the dominant members of the community. This disruption of our bacterial communities can make some of those nasty side effects even worse. Recently, our lab added a new piece to the puzzle. We discovered that harmful intestinal bacteria are capable of exploiting the cell death process for their own benefit. More specifically, the molecules that dying cells produce can actually be used as food by certain types of intestinal bacteria, and this enables them to become the dominant members of the community and actually contribute to these severe side effects. But what we don't still understand is which molecules are actually serving as food for these harmful bacteria. So therefore, we can't block this process yet. In our new research project, funded by Cancer Research Institute Ghent and the generous donors who contribute, we hope to identify the exact substances involved to be able to block their release in the future and thus prevent these harmful bacteria from becoming the dominant members. Eventually, we hope to limit some of the harmful side effects of chemotherapy and increase the quality of life of patients.